Okay. Well, thank you for joining in on my webinar here. My name is Michael. I want to talk about transcript assembly and RNA sequencing. My specialty really is in developing both molecular and bioinformatic protocols for getting the best picture possible of RNA at the isoform level. And so I want to thank LEXOGEN for inviting me to perform this webinar, and it will be sort of in two parts. First, I'd like to talk about how we got to the present day, sort of RNA sequencing technologies up to now, and then I'd like to introduce my own software package I've been developing and sort of the ideas behind it and how it performs. So here is an end-to-end rethinking of transcript assembly. That leaves us in a very interesting position because we have many different methods to capture RNA molecules, but it isn't totally resolved what actually is the best way to identify transcript isoforms from cells of interest. And so I hopefully can give you what I believe is a good answer to that question through the software that I've been developing. And so in order to think about how to best identify transcript isoforms from cells, then I was specifically interested in three different aspects. I really wanted to know, does including RNA end information, basically experimental evidence from RNA 5' ends and 3' ends, where they start and end, does that help you build a better annotation of transcript isoforms? I was also interested in the diversity of RNA sequencing methods and whether it was possible to combine or integrate different data types in order to build a better assembly. Particularly, I wanted to know whether short reads and long reads could be used effectively together in the same assembly model. And finally, what I was really interested in was whether it was possible to build a reasonable assembly of transcript isoforms from single-cell sequencing experiments. And so these three questions, I'll start with the RNA end information, and that single question was the seed that really started my development of the Bookend software platform. And so this was published over the summer, and I'd encourage you to check it out. But the essential concept is taking the 5' and 3' ends of RNA and using them to build a better assembly. But in order to do that, you actually have to have experimental 5' and 3' ends. And when I took a closer look at some single-cell sequencing protocols, this is an example of what's called SmartSeq2. It utilizes template switching onto what's called a template switching oligo that I'm showing here in blue, that after reverse transcription, the RT enzyme produces untemplated cytosines. Those can hybridize with a template switching oligo in solution, and you produce essentially an oligo tag at the 5' end of your cDNA. And then it's clear from the protocol as it's shown here that the tag at the 5' end and also the oligo DT tag at the poly A tail, neither of those tags are lost when the sequencing library is produced. And so from this single cell data, we should actually have a certain subset of our reads that are end labeled, that are either indicative of an RNA 5' end or an RNA 3' end. To test this, I generated SmartSeq2 libraries from 5 nanograms of total RNA. So this is a low input sequencing method, and just asked the question, can I identify the template switching oligo and the oligo DT sequences, and what percentage of those reads can I find them? And so it's not a lot. A very small minority of the total library contains these tags, However, when they're aligned to the genome and compared against known genes, they do concentrate at the transcription start sites for the start tags and the polyadenylation sites for the end tags. So this is encouraging in telling me that there's inherent end information contained in this single cell sequencing method. Also of note here is that I'm distinguishing between what I call start tags and cap tags. That's because one phenomenon of the template switching event is that if a 7-methylguanosine cap was present at the start of that RNA molecule, that is an indication that it was a full-length, intact RNA molecule. However, RNA is very fragile, and it quickly degrades both in vivo and in vitro. And one persistent problem in transcript assembly is RNA degradation, where you'll have a very dense representation of RNA at the 3' end, but as you go toward the 5' end of the gene, coverage drops lower and lower and lower. So I found that the presence of this cap structure actually produced a distinct tag from the template switching oligo, and I can separate 5' ends that are merely start tags. And so these could be template switching off of degraded RNA from those that genuinely have evidence of a cap. And we can see that the capped elements are much more concentrated at the transcription start site than the template switching events overall. And we can also see that these end tags, so the start and the end tags, are abundant enough that a majority of sequenced genes actually do contain at least one start and end tag. And so this was encouraging enough for me to sort of move forward and think about how ends could be incorporated into an assembly algorithm. And so without going into exhaustive detail of the algorithm itself, the essence here is that the genome is broken up into genomic fragments or regions that overlap observed reads, and they are divided up based on different border sites. So a 5' or a 3' label acts as a boundary. And you can see the boundaries here in red and green, and also splice donor and acceptor sites. And so when broken up in this way, then you can condense any RNA sequencing library into this sort of dense representation that I call the membership matrix, where each element in the membership matrix either includes or excludes different fragments of the genome. That means that different reads are either compatible or incompatible with being part of the same full-length RNA as other elements in the matrix. So this matrix can then be used to build an overlap graph, a condensed version of an overlap graph similar to what was used in cufflinks. And paths through this graph merely need to connect a start to an end. And I do so in a pretty simplistic, greedy algorithm that basically takes the highest weight elements, the ones that have the most evidence, the most RNA coverage, and I work my way down, building only complete paths. So complete paths connect a start to an end and have no gaps in between. And using these algorithms, I built out an assembler that can produce annotations from end label data. So if we take a look at this floral bud data and first compare it to the reference annotation, then I can see that by including end information, I'm both able to reconstruct more reference transcripts than if that end label or those end labels were ignored. But more importantly for me, the precision increased spectacularly. So merely including evidence of where RNA starts and ends allowed me to not construct 43% of the non-matching transcripts. These are transcripts that have no analog in the reference. And so in this case, I'm assuming that these are artifacts, are falsely assembled transcripts. And compared to existing transcript assemblers, we see that not only can more correct transcript isoforms be assembled, but that I am removing anywhere from 2/3 to 84% of the artifacts that are produced by these traditional assemblers. So at first glance, it really does look like end information plays an extremely valuable role in transcript assembly. Now, I mentioned that we have other ways to validate these annotations and assess whether or not they're good. I wanted to actually zoom in on the 5' and 3' ends and understand how accurate they were on a nucleotide level. And so here we have CAGE data sets that capture just transcription start sites. And I'm showing the nucleotide enrichments, so C in blue and then A, T, G. And we see that around known transcription start sites defined by CAGE, we have very characteristic motifs of Arabidopsis transcription start sites, including the Tata box, which is universal in eukaryotes, and then the pyrimidine-purine motif at the +1 position. And there's also this pyrimidine patch, the Y patch, that's shown as an enrichment of C. Similarly, direct RNA-Seq can show us what the nucleotide enrichment around 3' ends, around polyadenylation sites, looks like. And if we compare the ends of my annotations built by Bookend, we can see that it fully recapitulates the motifs seen at 5' and 3' ends, not just for sites that we know about. So these are 5' and 3' ends that are present in the reference, but also if we subset for just novel 5' ends and novel 3' ends, those motifs remain intact. Whereas if we look at another assembler, for example, string tie 2, then the nucleotide preference around these sites is essentially incoherent. It looks as if novel 5' and 3' ends are largely artifactual in other assemblers. And I also wanted to actually directly compare these results to long-read sequencing, because we know that with, for example, PacBio, you sequence the molecule from end to end. And so you can look at how well the full exon chain is reconstructed. And so notice here the all bar is the number of assemblies in each method that had at least one PacBio read fully supporting it. So from end to end and all of the splice junctions. And we can see that string tie 2 performs the best, but only a little over half of its transcript isoforms were supported by PacBio reads that were generated from the same tissue. And if we compare that to Bookend, we get about an 80% validation rate across all transcripts. And it's noteworthy here that even the isoforms that are not present in the reference, things that have novel splicing, novel starts or ends, or are antisense to the reference, still had some support from the PacBio reads. Again, there's a caveat here that that PacBio sequencing, especially at the time that this was performed, is much lower throughput. And so if novel transcripts are rare, then the validation rate will be lower if your throughput is low. So from these first pilot experiments, can I answer the question, does RNA-N data improve assembly? I think unequivocally, yes, it does. And now I was interested in what this end-guided model was capable of. And so if we think more about how to incorporate, say, long reads into an assembly, then we can see that the splice graph really causes us to run into some problems. Let's illustrate what these molecules look like. We have two of one isoform and two of another, where this alternative acceptor site is present in the lower two molecules, as well as an intron retention event. We can imagine if we build a splice graph from these four molecules, that you could draw a path that goes from A to C to D to F. That's a path that represents this top isoform here. You can also build a graph or a path from A, B, C, D, E, F, and that represents the lower molecule. But there's nothing stopping you from also drawing alternative paths through the splice graph that don't represent real molecules. And so there's a danger if you are incorporating long read data into your model that the information from different parts of the transcript sort of get broken up. And I don't want to do that. I want to keep the full chain of information intact. And so that's what the membership matrix can do, in that I represent each read as an element in this graph that has either includes or excludes every one of these genomic fragments. And so we can immediately see that there is no way to combine reads that represent element 1 with element 2 here. They are simply incompatible, and they represent entirely different isoforms. They are distinct from each other. So if we take a look at all RNA sequencing data types, we can see that the membership matrix generalizes very well, and very simple sequencing methods. So unstranded single-end short reads, they simply produce low information content elements in the matrix. In contrast to that, we can see that long reads will simply be full paths in the graph. So if they connect a start to an end, and specifically a cap to an end, then they represent a full path in the matrix, and there's no chance of them being misassembled. So if I directly compare these two libraries, the SmartSeq and the PacBio, the long reads, then we see that there are some differences in how they were generated, and the total depth and cost varied quite a bit. So at the time that this was performed, the cost per base was much, much higher for PacBio sequencing, but it is improving every day. And if we look at how long reads align to the genome, we can see that we do get full-length reads from start to end, from cap to tail. But we also see this sort of cascade of degraded RNA that here we can identify as lacking a cap based on its template switching oligo sequence. However, standard methods for summarizing the PacBio reads end up misidentifying this cascade of degraded RNA as alternative transcription start sites. And so by being aware of the cap structure inherent in the data, Bookend can avoid assembling transcript artifacts that are simply degraded RNA. So overall, if we pipe long read data through Bookend instead of the standard software, then we can see that we recall a much better collection of reference transcripts while assembling fewer artifacts. If we layer short reads then on top of the long reads to generate hybrid assembly, then the sensitivity goes up even further. And so it looks as if there's no reason why long reads and short reads cannot be integrated into a unified model of transcript annotations and be used together in assembly. And finally, this was the thing I was really interested in, is how low can you go? Can you actually get an accurate transcript assembly from single cell information? And you can imagine that, that if you look at a bulk RNA sequencing data set, then you have a pile of reads on a gene. But, and that could be represented by a single isoform. But if you actually look more granularly at the single cells that make up that tissue, then you might see a different picture. It could be that distinct cell types are actually producing distinct transcripts that can't be parsed apart from each other if you're looking at the bulk data. And so the isoform diversity might actually be underestimated if you're not looking at the single cell level. So interested in this question, there was a spectacular benchmarking data set that was generated by Sarah Teichmann's lab. And this was a plate of 96 mouse embryonic stem cells in which they introduced both the ERCC spike-ins and the SERV spike-ins that I mentioned earlier in this talk. Again, to reiterate, the SERVs are really a simulation of transcript isoform diversity. And so they're an extremely challenging set of isoforms to reconstruct from short reads. I tried to do it anyway. So if we take a look here, the ground truth in the SERV spike-in mix E2, this actually has all 69 molecules, but present in different quantities. So some are more abundant than others. And you can see that the dark green is most abundant, followed by the green, the yellow, the orange. In attempting to reconstruct the SERVs from each of the 96 samples, each of the 96 is shown as a single column in this bar chart. I can see immediately that that Bookend does a much better job of reconstructing the most abundant spike-in transcripts from this single cell data. And the less abundant transcripts are a little bit of a crapshoot still. They're very statistically noisy, especially when you're trying to amplify RNA from such a tiny amount of starting material. However, Bookend strongly outperforms in both recall and precision on the other transcript assemblers. And if we sort of extrapolate into the actual RNA from the mouse embryonic stem cells, then we can see that whether these isoforms match the mouse reference annotation actually shows extremely poor precision. And Bookend, on the other hand, since it's aware of the 5' and 3' ends, is far more precise. We lose some sensitivity because single cell data is very incomplete. It's very hard to find reads from the entire length of a transcript from a single cell. But this data can be supplemented through hybrid assembly if we can provide cage and 3p-seq data. These are the 5' and the 3' end information. If we provide it alongside the single cells as a hybrid assembly, we see that that sensitivity is restored. So really the loss in sensitivity can be attributed to the fact that it's harder to pick up this small number of 5' and 3' ends. But to show you the magnitude of the problem, this really stunned me when I first saw it. I am showing assemblies from single cells and then merging the data into 2, 4, 8, and all the way up to the full plate of 96 cells. And we see that that more correct assemblies, more ref-seq matching assemblies are built as you combine data from more and more cells. It does start to plateau though. However, the number of non-matches grows linearly with the amount of sequencing data you add, to the point where other transcript assemblers are actually building 10 times as many false matches as true matches. And you can just imagine how frustrating it would be to sift through an annotation where 9 out of 10 transcripts is likely an artifact to find any sort of meaningful data to follow up on. So here I'm just illustrating at a single gene what it looks like to be as precise as possible in performing hybrid assembly. So it's possible to take the single cell data sets and actually assemble on the single cell level and then assemble the assemblies. So I'm showing here 96 individual assemblies that have been condensed into one large pile. That pile is passed through Bookend a second time in order to assemble the assemblies. If we layer in the 5' and 3' data from other experimental methods, then we can build one giant meta assembly. And from this meta assembly, we can see that not only are certain reference annotations, such as gen code, somewhat fragmented in the isoforms that they show, particularly the low confidence isoforms, but also that the reference annotations are quite incomplete. So REF-seq does not contain this alternate poly-A site that dominates in mouse embryonic stem cells. And that can really be important to know if you're attempting to quantify, say, tissue-specific transcripts. And to take a closer look globally at this, this idea of meta-assembly, of assembling the assemblies, then we can look at performance metrics, recall and precision, from these different assemblers. And when we meta-assemble, for example, with Taco, this is a multi-sample assembler. It was published in 2015, I believe. The sensitivity goes up marginally. You can see that from the base assembly to the meta assembly, we gain a little bit in precision, but lose a lot in sensitivity. And in fact, compared to the Bookend base assembly, the Taco meta assembly loses both a dramatic amount of sensitivity and precision. If we take the base assemblies from any initial assembler and pass them through Bookend a second time, then we can see that we're actually getting a much more true to the reference picture of transcript isoforms. But you may have noticed that this y-axis doesn't go to 100 and that there are fundamental limitations to short reads. And so I was showing you that, yes, Bookend does assemble more true spike-in molecules and fewer false serves than other assemblers, but it's still, there are 69 molecules in this data set, and you'll see that there are almost as many false assemblies as true assemblies. And I think this really reflects sort of a wall that you hit with short reads. The short reads are fundamentally missing long distance information. And you have to basically guess about long distance connections in a transcript using statistics. And so if we take a look at, say, the spike-in molecule SERV5, we can see that there are events, there are variations at the 3' ends or at the 5' ends that are far enough apart that no short read, which may only be 200 to 300 base pairs, could possibly capture. And so that brings up the possibility of assembling long read sequences. And another form, I showed you PacBio reads before, but this is long read data on the SERV molecules from this paper that came out in the spring, just a month or two ago. And this is Oxford Nanopore direct cDNA sequencing of the SERV molecules. And you can see that even though it is quite noisy at both ends, that there is long distance information that's present in long reads that just fundamentally doesn't exist in short reads. And so passing Oxford Nanopore reads through Bookend really does recapitulate transcript isoforms in a way that simply isn't possible with short reads, unfortunately. And if we look at the ONT reads across all SERVs and ask how many can be reconstructed by both StringTie2, by IsoQuant, which was published here with this data set, and passed through Bookend, we can see that we're still not at 100% sensitivity and 100% precision yet. However, I think it's clear here that that Bookend is much more accurate at getting the precise 5' and 3' end of the molecules and assembling a closer to true picture than we've been capable of getting so far. And so to summarize sort of the points that I was hoping to make today, then these were the 3 questions I was ultimately interested in asking. Does RNA-N data improve assembly? I think we can say unequivocally yes. Short reads and long reads, are they actually useful together as a hybrid assembly? I would also argue that yes, mixing modes of sequencing allows you to make up for deficiencies in either sensitivity or information content that other sequencing methods lack. And finally, is single-cell RNA sequencing valuable for annotation? I think the answer is yes, with an asterisk. It certainly does seem to be the case that transcript annotation on single cells is possible, but with short read methods alone, there are fundamental limits to the precision of the annotation that can be built. So long read single cell methods are coming, and they are on the horizon, and I'm very optimistic for those. So thanks for paying attention to what I had to share today. This research was performed in Vienna, Austria, at the Vienna Biocenter in the lab of Michael Nodine. So this is his lab back in, it might have been pre-pandemic, 2019. And since then, you can find both Michael Nodine and myself at Wageningen University here in the Netherlands.
Hello, and welcome to this short presentation about LEXOGEN spike in RNA variant controls. My name is Renata Kovarik, and I am a product manager at LEXOGEN. LEXOGEN is the RNA experts biotech company. We are a group of scientists with a passion for RNA and a strong will to serve the scientific community to drive research progress. LEXOGEN was founded in 2007 and is based in Vienna, Austria. We at LEXOGEN develop our kits and services to empower our customers with innovative solutions for RNA research and transcriptomics. LEXOGEN offers kits covering the whole RNA-Seq workflow, from sampling, RNA preparation, library prep, to NGS data analysis, and also NGS controls. Although we routinely include even several controls and standards to our experiments, using controls in RNA-Seq workflow is not so common, despite of the fact that biases might be introduced during each step of RNA-Seq workflow. Therefore, LEXOGEN offers external spiking RNA variant controls, or SERVs in short. SERVs are artificial RNA controls which allow to validate the capacity of the RNA-Seq workflow to detect isoforms, quantify them, set up and validate experimental and pipeline parameters. SERVs also allow better quality control of RNA-Seq datasets obtained from different labs or over time. SERVs controls are added during or after RNA extraction, and they are processed together with RNA-Seq samples. Usually, only 1% of total reads is dedicated to SERVs. Even this little fraction is sufficient for validation of RNA-Seq workflow and assessing the performance and the limits of the given setup. SERVs are artificial sequences. Therefore, they are universal and they can be used with any sample type or species. SERVs are also polyadenylated, so your library prep can start from poly-A selection, rRNA depletion, or total RNA. Transcriptome has high complexity with large concentration differences and contains several RNA classes with specific properties. SERVs were designed as a family of modules where each module mimics a specific aspect of natural transcriptome complexity. SERV isoform module probes transcription and splicing variant. ERCC addresses concentration dynamics, and long SERVs mimics length complexity. SERV isoform module mimics isoform complexity by 69 isoforms that map to 7 different loci, including antisense transcripts. They are derived from human model genes, but they have unique sequences with no significant similarity to any known sequences. This module probes alternative splicing, alternative transcription start and end sites, overlapping genes, and antisense transcripts. This allows you to test detection and quantification of isoforms in your RNA-Seq workflow. ERCC module contains 92 transcripts that follow one gene, one exon, and one transcript layout. ERCC module assesses a dynamic complexity of transcripts concentration with a range of 1 to 1 million. Therefore, it enables input and read-depth-dependent detection and quantification, and also straightforward input-output correlation. This module serves as a standardized assessment for dynamic range, dose response, lower limit of detection, and fold change response. Transcript of SERVs and ERCC modules have an average length of 1 kb, which does not cover the long transcripts. Long SERVs module contains 15 transcripts with length of 4, 6, 8, 10, and 12 kb, with 3 transcripts for each length category. Long SERVs have 1 transcript per gene, so similarly to ERCC, they do not have isoforms. Spiking controls are essential for RNA-seq experiments. They allow validation and monitoring of the RNA-seq workflow and assessing technical parameters such as bias, sensitivity, and length representation, and also evaluation of functional aspects like isoform detection and quantification. This allows you to know the performance and the limits of your RNA-seq workflow and data evaluation pipeline. Moreover, with spikings, you can evaluate comparability of samples in experiments, but also between experiments. Therefore, you can evaluate technical variability between samples in experiments before investigating biological variants. If you need help with selecting a proper serve set for your experiment, please contact us at support@lexogen.com. If you're interested in learning more about serves or any other LEXOGEN products or services, please check our webpage, lexogen.com.